All right, so we're going to do two things today. First of all, I'm going to talk just very briefly about how to access data sets for use with Horos, so MRIs and CTs and other kind of medical imaging data sets. And then I'm going to actually demonstrate how to work in Horos. And one more thing I got to do here. I got to make sure that I'm sharing my display properly. There are two places where we can access data to use in Horos. One of them are the old Osirix data sets that I showed you briefly before, which we can access directly from the course share. So if I go to my desktop and click go, say connect to server, it just conveniently enters the address that I had there last time. If you don't have this address, I mean, I suppose I can leave it up there for a minute so you can try to read it and copy it. It's also included in the course syllabus on my courses if you want to access it that way. I'm going to go ahead and click connect. It's going to want my RAT password. So then you can either pick 2245, which is the current semester, or just click current. Find my name. And then I have 3 courses in here. I think probably you'll only see one, whatever course you're registered for. But I'm going to open up the public folder. And there are 2 main folders in there. There's one called sample PDB and mole files that we'll come back to later in the semester. And then there are a couple folders of medical imaging data sets. And so we've got the list of Osirix and Horos data sets. These are the ones that all have these strange names that end in IX. We go back into a web browser and we go to osirix-viewer.com, or that link is also, there's a link directly to it here in my courses. On the Osirix page, you click on resources and then DICOM image library. And this is where you can preview these different data sets. Now, obviously, I don't have all of them, but there are a handful that I have. This one called Anonymized 2, that's actually the one that I've called Canine Thorax because it's a dog. Oops. Do that. You know, carcinomics, that's one that I have. Seed automatics, colonics, anyway. So you can preview them here just to get a sense for what part of the body it is and what kind of imaging technology it is, whether it's CT or MRI or something else. And then from the courses share, you can simply grab a couple of these and drag them onto your desktop, copy them off. I'm going to grab just a couple of them so that I can use them to demonstrate for you. It doesn't matter which one you use. In fact, I encourage you when we get to the exercise part of the class, I encourage you to explore a couple of them, just to see what kind of body parts are included in each of these data sets. That one and one more. Seems to be running incredibly slow today. Usually downloading things off the courses share is really zippy. Even though these are pretty big data sets, they're a couple hundred megabytes. Usually it's very quick. Maybe it's because you're all simultaneously trying to access them. All right, so that's all I'm going to grab for the moment. So that's one source of data. Another source is something called the Cancer Imaging Archive that I talked about very briefly in the previous lecture. That link is included right in my courses for you. So here's the Cancer Imaging Archive. And the way you use this site if you want to retrieve some data to play around with in Horos, is you're going to click on the link called Access the Data and come down here to where it says Browse Collections. And we scroll down, we see that there's a long list of different studies, and it's showing 1 through 10 out of 218 different entries. These are, different kinds of cancer from different regions of the body. You can actually click to sort these by region. So that sorts them alphabetically by region. So starting with abdomen and then moving to things like adrenal. And of course, there's numerous pages. Whoops. Not sure what happened there. That's weird. So let's see. You can also sort by cancer type. So acute myeloid leukemia, adenocarcinoma of the colon. That sounds like a promising one. So once you find one that interests you, either by body region or by type of cancer, you simply click the blue link at the far left, the code number, and it brings you to a description, a summary of what's included. And then down near the bottom, there are a couple of buttons here. There's a blue button to download the entire data set, and then there's an orange button that allows you to search within the larger data set. I don't recommend clicking on the blue button because oftentimes these are enormous collections, many gigabytes of data. So you probably don't want to click the blue one. Instead, we're going to click search. Again, for some reason, things are running really slowly today. But what this allows you to do is to narrow things down by different kinds of imaging modality, body region. Okay, that's weird. Species. These are all mouse. All right, so that was a really bad choice. I don't know. That could be fun. Look at some mouse cancer. So I'm going to narrow it down by just MRI. You can click where it says more here, but it looks like all of these are zero. So there's 24 different data sets of MRI. They're all from the abdomen. All 24 of them are from the mouse. So, not necessarily what I wanted, but what the heck? Let's take a look at it anyway. Now what you see here is a list of collection IDs and subject IDs. Each of these was actually a different study, a different imaging study, apparently on a mouse. And there's 3 pages of them. And you can't really tell much from just looking at these numbers. You can click the little triangle of the left that might give you a little bit more information. It tells you the day, tumor characterization. There are 2 different series in there, meaning, this could be like a CT and then some other kind of scan, like a PET scan, or it could be a T1 and a T2 MRI, something like that. It's really hard to know exactly what you're getting with these. But once you've identified some studies that you think might be interesting just to look at and play around with, the way you retrieve them is really kind of awkward and strange, in my opinion. It's like shopping in Amazon. You put things into your cart, and then you have to check out of your cart. So there's literally a shopping cart or shopping basket icon here. So I'm going to put a couple of these in my shopping basket. And then we go up to the little shopping cart icon in the upper right. And we see these are all of the individual studies that I've put into my cart. They were part of those 2 larger data sets. And then I want to download them. There's a download button up here. And even this is awkward. You'd think it would be nice and straightforward. In order to download something from the Cancer Imaging Archive, you actually need a separate app installed on the computer called the NBIA Data Explorer, I think is the name of it. And I'm crossing my fingers that it's still installed on these computers. So I click download, and it downloads this thing called manifest, then a long number, TCIA, or TCI, yeah, TCIA. I'm going to click to open that file. And all right, thank goodness the NBIA data retriever is installed on this computer. So it brings up this separate data retriever window. You agree to their terms. And then it brings up this window, which again lists the different data sets that I'm going to retrieve. At this point, down near the bottom, you want to click the Browse button, because what that's going to do is it's going to just allow you to tell the data retriever app where to put the files. So I'm going to click Browse. I'm going to say, let's put them on the desktop, but I'm going to put them all in a new folder. So I don't clutter my desktop up too much. Create that new folder, click Open. So now it knows where to put the files. Then I click the Start button at the very bottom, and it'll show you the progress up here of downloading those data sets into that folder. Right, and then when we get into Horos, we can drag these data sets into Horos and work with them. Okay, so that's complete. So we can close the data retriever. I'm just going to go ahead and exit out of the cancer imaging archive at this point. Now, it used to be that I would show you a third source of imaging data. The Visible Human Project had a set of CT data. I think I put the link in the syllabus, but when I went to the link over the weekend to double check, I couldn't access the data. It seems like every year that I teach this class, they move it around somewhere else. So I'll try to search for it and see if I can figure out where they put it this time. But for the time being, we can't access it. So I'm not gonna worry about it. All right, so we have a few different data sets here. I have a couple that I grabbed off of the courses share, one called Mannix and one called Mechanics. We've got a few data sets here that are all from the Cancer Imaging Archive. And just for fun, this is my MRI from a couple of weeks ago. I was there on Tuesday for a follow-up with a surgeon. And while I was there, I said, hey, any chance I can get my images? And they were like, oh, sure, just go downstairs and ask them for a disc. And so they handed me a disc full of DICOM files. So I'll show you, I get to show you my MRI. All right, so now we are going to open up Horos. So I'm going to go to the Applications folder, launch Horos. And here's what Horos looks like. So we start out with what's called the database window, which is where we can view all of the different data sets that we've imported into Horos. At this point, of course, there's nothing in there because I haven't imported any data sets in yet. There are three different ways of importing data into Horos. You can go to the File menu and choose the Import command. You can simply click the green arrow that says Import right here, or usually the easiest way to do it, what I prefer to do, is just drag the folders and simply drag and drop them into the application. You generally want to choose this command that says Copy Files, which makes a duplicate of the original files. Put my MRI in there. And then the Cancer Imaging Archive, I'm going to go into these subfolders and drag those in. And so all these with the long numbers that start with 99, those are all the Cancer Imaging Archive files. So now we can see that we've got a whole bunch of different data sets that we can choose from. Within each data set, oftentimes there are many different studies. So there might be CT, there might be MRI, there might be multiple different kinds of MRI, like T1s and T2s. There might be MRIs with contrast, without contrast. There might be some PET scans in there or ultrasound or any number of things. So there's a little triangle here next to the name of each data set. I'm going to click that next to my MRI. And we see there's a whole bunch of things here. There's some coronal slices that are T2 weighted. If I scroll across, we see it's got my age in there. That's interesting. It's got a description of the study. So in this case, it says MRI of the spine with contrast. The imaging modality is MR, again, telling you that it's a MRI. One of the most important columns in this database window is over here near the far right, number of IM, that's number of images. So the number of separate images that make up that data set. General rule of thumb, more is better. If you're deciding, gee, should I look at this MRI or should I look at this other MRI? The one that's got 175 images is almost always going to be better than the one with 32 images. Just means it's finer resolution, more detailed. Not always the case, but usually that's a good idea. So if you look at mine, none of them actually have a really overwhelming number of images. There's an axial set of axial sections here with 54 images. There's some more axial sections down here with 76 images. Let's see, I'm trying to remember, I previewed these. Here's some T2 axial sections. It's only 36, but just you kind of preview it down at the bottom. These look like pretty nice, clear images with a lot of interesting anatomy in them. So let's say I want to dig into this a little bit more deeply and actually look at the images. So I've selected this axial T2 MRI. Up at the top, there's a little icon that looks like an X-ray with a magnifying glass. This is the 2D viewer window. I'm going to click there, and it opens this new window that allows me to look at all of the individual images, all of these individual two-dimensional images. So if I remember correctly, this is actually one of my thoracic vertebrae. This is like T12. It's one of the lower thoracic vertebrae. You can simply use the arrow keys on the keyboard to scan through them. So I can use the right arrow and just cycle through them. So I think we're getting down to L1. Just a little anatomy review. Let me get down to a good level here with some nice clear anatomy. All right, so that oval thing kind of near the top of the image, that's either part of the vertebra or part of the intervertebral disc. Any guesses what these other black blobs are immediately on either side of the lumbar vertebrae? It's a good, what is it? Nope, that's adjacent to the body. The transverse processes are going to be more posterior, so closer to the bottom of the image. They're muscles, right? Can anybody think of a muscle that would be round in cross section that sits on either side of the lumbar vertebrae? You want to announce to everybody what it is? Psoas major. All right, those are the psoas major muscles sitting on both sides of the lumbar vertebrae. And so back here, you can see the lamina of one of the lumbar vertebrae and then joining and then forming the spinous process, projecting posteriorly. You can also see the facet joints. So there's a black line that shows up on both sides, kind of a black line right here and right here. That's actually the joint between the superior and inferior facets of two of the lumbar vertebrae. Oh, you actually can see it better here. There's that the facet joint between superior and inferior facets. I'd like to try to show you some pathology in here if I can, but it's a little hard to see. So this, I've had lumbar surgery before for the same condition, for sciatica, and it's a little hard to tell on here, but there's a little gap in the bone right there. There's a little notch taken out of the lamina on the right side. So this is the right lamina of the T5 vertebra. So they actually did a laminotomy where they nibble away a little bit of the lamina to get at the nerves inside. And then now we're getting down into the sacrum. You can actually see the sacral ala here, the wings on either side forming the sacroiliac joint with the ilium. And then you can see my erector spinae muscles down here near the bottom. They're nicely marbled like a good steak of fat in my erector spinae. All right. I'm going to open up a different data set. One thing I know with MRIs, they don't reconstruct very well in 3D. And it's kind of hard to work with them. So the rest of the demo, I'm going to focus on CTs. I just realized that our mice are also MRI, so I may not really explore those because MRIs don't make for very good 3D reconstruction. So let's focus on these other ones. So let me open up a data set called Mannix, which I know is a really, really nice CT of head and neck. And once again, we've got 4 different data sets here. The people who made Osirix originally, and the people who made Horos also, they were from Switzerland. And so a lot of the stuff tends to be in French when you look at the data sets. So sans IV means without IV contrast. Avec IV means with IV contrast. Angio CT, you can kind of tell what that means. It's a CT, but with imaging of the vessels. So it's an angio, kind of an angiogram. If I look across to our number of images column again, all right, there's one with 460 images. That's our angio CT. So that's an incredibly detailed CT study of this person's head and neck. So I know this is going to give us some really nice images. Once again, I'm going to click the 2D viewer, and that will bring up the individual slices. So here we are down in the thorax. You can identify the lungs clearly on both sides as the big black areas. You can see the white areas in the middle of the thorax, which are going to be some of your large vessels like the aorta. And as I scroll through this, we're gradually working our way superiorly. So we're going from the thorax up towards the head. Now we're getting into the aortic arch. So you see the big white sausage there is the aortic arch. You can clearly see the thoracic vertebrae down near the bottom of the image with the articulation of the ribs. You've got the head of the rib articulating with the vertebral body and the tubercle of the rib articulating with the tip of the transverse process. And then as we keep scrolling through, eventually we'll get up into the neck and the head. So now the lungs just about disappeared. We're up near the top of the lung, the apex of the lung. That's like probably the T1 vertebra right there. Now we're getting up into the cervical vertebrae, into the neck. You can see here's a mandible starting to come into view, you know, his chin up at the top of the image. You can see the trachea, still nice black triangular structure. A couple of teeth. So now here's the upper teeth. And then there's his nose, nasal cavities coming into view. nasal concha up here in the nasal cavity. Now we're getting up into the skull. This dark space down here near the ear, that's the mastoid process. It's filled with little air cells, little sinuses. Oh, you can see his eyes now. We're up in the orbits. All right. So anyway, you can scroll through and have fun looking at the individual slices. But the really interesting part of Horos is the ability to then take those individual slices and stack them up to create a 3D model. So there are two basic kinds of 3D models that we can create in here. And your exercise today is going to have you create one of each. We're going to create what's called a surface model, and then we're going to create a volume model. I explained this briefly last time, but I wouldn't expect you to remember the difference. So with a surface model, what Horos does is it looks for a sharp boundary or sharp interface between pixels of very different value. So for example, in this image right here, you can see that all the bones are very bright white. And then they're surrounded by soft tissue that is all various shades of gray. So Horos could easily detect that boundary between the bright white bones and everything else. Or alternatively, Horos could detect the boundary between all of the skin and the soft tissue, and then the black outside of that, which is just air. And we can tell Horos which of those boundaries to identify: the boundary of skin, the boundary of bone, etc. So to create this surface model, we're going to go to a button up the top as soon as the little zoom menu gets out of my way. Come on. I wonder if I can. Up here, there's a 2D, 3D button. When I click on that, there are a number of choices here, but the only two that we're going to work with are 3D volume rendering and 3D surface rendering. So I'm going to choose 3D surface rendering. And I can choose a maximum of 2 surfaces. Right now, there's only one surface checked. It says first surface. It wants me to enter a pixel value, which somehow corresponds to the brightness of the pixel. Honestly, I've never been able to figure out exactly what the numbers mean. I wish they had them as like simple, like an RGB scale, or something like from 0 to 255, which is your standard grayscale or RGB. So I don't know exactly what the numbers mean, but the nice thing is Horos includes some preset values. So I can have one, I could say CT skin, which is going to recognize that boundary between the skin surface and then the air around the outside of the head. Now that's also going to recognize the boundary between things like the trachea and the air inside the trachea and the lungs. I can give this a color if I want. When I click the color box, I could just, choose the color picker and, give somebody like a little light brown skin tone and click OK. And there he is. Now that I've got this 3D surface reconstruction, I have a few tools in the upper left that I can work with. Magnifying glass, which works pretty simple. You drag up and down to zoom in and zoom out with the magnifying glass. There's a rotate tool that rotates only in an axis that's basically coming straight out of the screen towards us. There's a translate tool, which just lets you move up, down, left, and right. And there's a 3D tool that allows you to rotate in any plane. So you can rotate, up and down, left and right, and so forth. And when I do this, number one, you see a very convincing likeness of this person's face. Yeah, question.
When you get to that menu bar on the top, I noticed kind of switched a little bit from what it was. Like from where we had all of the CT scans to where you hit the button to get to that page.
Right here?
Yeah.
Okay, so first thing I had to do, so from this database window, All right, I picked one of our data sets, which was this angio-CT, and then I click 2D viewer first, which brings up this window where we can now see the individual slices. And then from here, we can access the 2D, 3D button to open up surface rendering. Right. So like I said, we got just a few buttons. We can zoom in and out, we can rotate, translate, or we can rotate in 3D. And as I was saying, we get a very good likeness of this person's face. You can even see the jammies he's wearing or whatever he's wearing, and even the button on his shirt. And if we tilt him back this way, you realize that we're also imaging the inside of the airway. So we're seeing the inside of the trachea and branching of bronchi within the lungs. And we can even zoom way in there. If you zoom in far enough, you can actually fly right into him and go up and even explore the inside of the pharynx and the larynx way up high. In fact, if you go way in there, you see the things off to the left and the right, that tubular looking thing on the left side and the right side of the image. That's the ear canal. That's the external auditory meatus and the whole middle ear cavity, which again are also, filled with air. So that's a surface model with just one surface. Once you're in here, you can click the surface settings button, which is this grayscale face icon, and it'll bring you back to this window here. We can change to a different kind of surface model. We can look at only the bones as a predefined value, and that enters a different number here for pixel value. And then I'm going to just change my color slightly, get more of a bone sort of color. Click OK. So now it finds the interface between the bones and all of the surrounding soft tissue. Now I think you can tell that it's kind of ugly. The vertebrae are all fragmented. It's just because of the numerical value that was automatically entered, that 500 number, it's mistaking some of the bone for soft tissue. It's like having a hard time to figuring out where that boundary is between bone and soft tissue. The skull looks pretty decent, but the vertebrae are terrible. You can go back in here and now start playing around with that numerical value, but I'll be honest, I've had a heck of a time trying to get it to really do much of anything. Like I just changed from 500 to 200. Let's see if that improves things at all. You'd think it would be straightforward, but it's definitely not. So that didn't seem to do anything. Maybe very slightly better. So you can just kind of keep doing this by trial and error and see what happens. The numbers range from like negative 500 to positive 500. So I keep reducing the number and really not getting a whole lot better. But even though that's kind of ugly, let me try one other thing. Go back to the surface settings. Let's try two different surfaces. So we've got our first surface is that bone, although I messed with the pixel value. We're going to add a second surface. This time we'll add the skin. So we can do both bone and skin. I'll choose a little bit lighter skin value this time. And you can also change the transparency. So I can make that skin layer partially transparent so that the bones will show through. Well, here we go. So we've got the skin and the bones. And you can also see, it's not just the skin on the outside, but you can see the trachea going up through the neck and up inside the head. You can see some of the nasal cavities and so forth. So we get some interesting, kind of cool stuff. So your exercise today is to create a surface reconstruction that has two different surfaces, and bone and skin seem to be the ones that work the best. And simply export this as a JPEG. I'm going to actually grab the window by the bottom right and just kind of drag the sides of the window in. So it's, so I'm not getting so much black space on the sides. If I had left the window the full width of the screen and I exported a JPEG, it would just give me all that excess black on the sides, which isn't really a big deal. But to export this as an image, cross your fingers that this works right, because last year for the first time, I was having all sorts of issues with this. Hopefully we won't have a problem with it. But I'm going to go to the file menu, say export, and say export to JPEG. And you can name it whatever you want. This is the Mannix data set. So I'll call it Mannix Surface. Put it on the desktop. Click save. Now here's the moment of truth. Let's see if this works right. Crap. All right, it's doing the same thing it did last time. So even though I could see the whole head in my preview window, when I exported the JPEG, it gives me just a corner. It's cropping off the image. And as I recall, last time we had to do some really weird workarounds involving the Wacom tablet and the screen settings. It has to do again with this whole mirroring thing, where I'm looking at one screen, it's mirroring that image up on the projector, and I think it's also trying to mirror it to the Wacom. I don't even know if the Wacom's working. All right. Sorry, it's really frustrating because this works perfectly at home every time I prepare, for this class. But that's because I don't have a Cintiq. I just have an old-fashioned Wacom drawing tablet. So I only have the one monitor at home, so I never have issues with mirroring or anything like that. I'm gonna make the Cintiq the main display. And the monitor I'm gonna mirror the Cintiq and projector. All right, that seems to be working. Hopefully this isn't screwing up the folks on Zoom playing musical windows here. All right, so I'm seeing the same image on my main display and on my Cintiq, but the Cintiq is actually functioning as the primary display. Let's see what happens now. I think this is the workaround we found last time. Okay, so now screen is black on Zoom. Okay. Charlie, are you able to see anything on Zoom now? All good. Okay, great. Thank you. All right. Hopefully you'll be able to continue to see what I do as I open new windows. But all right, so in this one particular data set, I'm choosing this angio aorta. So it's an angiogram. So it's a CT, but with contrast to see the vessels. clearly, and it's at 1.5 millimeter intervals, meaning the individual axial cross-sectional slices are only 1.5 millimeters apart. So very, very fine resolution, 743 images. So once again, I click the 2D viewer. You can see the individual images. Oops. Let's scroll through those. And now to create a 3D volume rendering, just go to the little gear icon and pick 3D volume rendering. And we see now, in this case, the entire thorax and abdomen, or most of the thorax and abdomen, even down into the pelvis. We've got a couple new tools. We've still got our translate tool that allows us to just move up, down, left, right. We've got the magnifying glass to zoom in and zoom out. We can rotate in one axis. We've got the 3D cube that allows us to rotate in many axes. We'll come back to the scissors and the little skull another time. But the tool that really distinguishes 3D volume rendering from surface rendering is this tool at the far left, which looks like a little grayscale from white to black. This is called the window level and window width tool. And at some point, I'll try to explain exactly what that means. But put it simply, this is a way of adjusting the transparency of different tissues based on the original shade of gray that they were in the CT or MRI study, in the original imaging study. I think I forgot to mention this with the surface model, but here in the volume model, as Horos brings the data in, it analyzes all the different pixels and says, okay, there's some bright white pixels, there's some solid black pixels, there's some intermediate gray pixels of different shades of gray. It automatically assigns a color to each of those shades of gray. And it says, okay, we think this intermediate gray is probably muscle, so we're going to give that red. The really light gray and white, that's going to be bone, so we're going to color that yellow. So it applies this default color scheme to the image. That's why when you look at this, you see things in various color. The bones are kind of white. There's some fat, which is kind of yellow. The skin on the surface is kind of reddish, pinkish. And what this window level and window width tool does is it allows you to then take certain values of pixels, like certain shades of gray, and completely hide them, make them transparent. Or it allows you to kind of shift the colors around so that the colors are assigned to different tissues. You have to be really delicate with this tool. Essentially, what I'm going to do is I'm going to demonstrate that you simply can drag it left or right, or drag it vertically up and down. And each way you drag is going to have a somewhat different effect on the body. Well, what you find is if you make a really big drag, you drag it very quickly across the page, all hell breaks loose and the screen just goes nuts, and you just see a bunch of static, or alternatively, your image just completely disappears. So generally speaking, you want to make very small moves to adjust these things in a very controlled way. So watch what happens when I drag slightly to the left. The skin is disappearing, and we're actually seeing some of the musculature. Like you can see a really nice rectus abdominis muscle down here in the abdomen. And then drag a little farther to the left. The colors are also changing. We're getting more of the yellow and white. And drag back to the right. Put some more of the skin back on. And I drag vertically now, and I drag down. And we're putting all of his skin back on so we can clearly see all of the skin. If I drag up, the skin disappears, subcutaneous fat disappears. eventually even the musculature disappears, and we're left with bones and arteries. The arteries are visible because they'd used a contrast material in the arteries. We can drag left and right again and change the colors of these things. We can drag to the right and it puts a little bit more color back on. Honestly, sometimes it's a little bit of trial and error. It's hard to know exactly which way you need to drag to get the different tissues to appear or disappear, and to know exactly how to apply the different colors. But just play around with it a little bit. Just try dragging up and down, left and right. You notice that I can actually make things like the kidneys visible. That's because the kidneys are very vascular. So the dye that's in the aorta and the other vessels is also dyeing the kidneys. I can zoom way in here and rotate around and actually start to see a whole lot of interesting anatomy. I've got the little hard to see the celiac trunk, but it's right there. And there are a couple of branches off the celiac trunk. There's the left gastric artery. There's the splenic artery going over towards the spleen on the left side of the body. This is the common hepatic artery, the gastroduodenal artery going down. And especially from the side, I can now see the superior mesenteric artery going down towards the intestines, and the left and right renal arteries supplying both kidneys. You can see the aorta down here bifurcating into left and right common iliacs. You can see the common iliac on the left side of the body splitting into external iliac, which is going to cross into the thigh, and then internal iliac, which remains in the pelvis. There's the superior gluteal artery exiting the pelvis through the greater sciatic foramen. If I rotate the body around, we'll probably see it coming out. There it is. There's superior gluteal artery right there coming out of the greater sciatic foramen. And you can clearly see the bones, really nicely, different parts of the bones. Right, way to do that. You can see spinous processes of the lumbar vertebrae, transverse processes. You can see the overlapping of the facet joints, which is something I was showing you on my MRI. Now besides just changing the colors and opacity of these different tissues, some other really cool things you can do here in these volume renderings. One thing is cropping. There's a crop tool. Let me zoom out a little bit more so you can see this better. Whoops. So there's this tool that's labeled crop tool, and it's this little cube with a bunch of little green tennis balls on the outside of it. When I click that, it brings up this bounding box surrounding the body and has those little green tennis balls on it. And those correspond to different planes like coronal plane, sagittal plane, axial plane. And we can click on each of those planes and the little tennis balls get bigger. And that means they're active. I can drag this tennis ball in and crop into the right side of the body in the sagittal plane. I want to move this around again so I can show you some of the anatomy that's visible, but I want to be careful. I always want to turn the crop tool off before I start moving around. Unfortunately, what happens if I were to try to move around now, like using the little 3D rotate tool, it's actually going to rotate the cropping box as opposed to rotating the anatomy. So I'm going to turn off the cropping box, and then I can actually rotate around and do things like zooming in. So basically, by cropping the right-hand side, I was able to just remove all of the ribs to get the ribs out of the way. So now we can more clearly see, the vertebrae and the aorta and some of the branches of the aorta. And if I want to uncrop, it's just a simple matter of just turning the crop tool back on. Just grab that tennis ball on that side and drag it back out. And you can crop in the other planes. You can crop in a coronal plane, cut away the front of the body. You can crop in an axial plane. That would allow us to, then look down on the vertebrae from above. Crop that. All right, so that's the crop tool. One last thing that I want to show you is there are these two buttons here. The label at the bottom says CLUT editor, and there's an 8-bit and a 16-bit option. CLUT is short for color lookup table. And that is what Horos uses when it decides that, okay, we're going to make the light pixels yellow, we're going to make the intermediate pixels red, we're going to make darker pixels, some other color. It's basically just a table that matches up each shade of gray with the corresponding colors that are assigned to those different gray pixels. And by opening up this thing called the color lookup table editor, it lets you customize what colors are assigned to those different gray values. And that can be really, really powerful in helping you distinguish tissues that may be very close in value to one another. You can make some very subtle little tweaks to the coloring and even to the transparency of different pixels in the image to make some things more obvious than others, make some things stand out better than others. So when I click the 16-bit color lookup table editor, generally we use the 16-bit one. It just gives you more choices in terms of colors. What happens is the image up the top gets a little smaller, and we get these little graphs down at the bottom. And there are two different graphs down here at the bottom. In the background is a grayscale graph, it's a histogram, meaning that the height of each part of that little graph, that little grayscale mountain range in the background, the height of the line is telling you something. The left-hand side of the image corresponds to the darkest pixels in the image, which would be things like the black air that's all around the body, as well as the inside of the lungs, which are going to be very, very dark. The far right-hand side of the image represents the lightest pixels in the image, which are going to be your bone, tooth enamel, or any metal that might happen to be in the image. And then all of the stuff in between represents the midtone values, which are all your different soft tissues. And then the height of that line in the background simply tells you how many pixels do we have in each gray value. So way over at the far left, we've got a big high peak telling us that there's lots and lots of black pixels. Not surprising because there's a lot of black airspace around the body. Then there are fewer in this kind of dark area. There's another peak here right in the middle, which probably corresponds to the muscles and a lot of the internal organs. And then it starts to trail off towards the right as we get into the bones. Um, you can, and then the colored graph tells you which colors are then assigned to the pixels of those different gray values. And notice right now that um, the colored graph only spans a relatively small portion of that total grayscale space in the background. Um, but we can then grab these little dots and move things around. I can grab the white dot and drag it all the way to the right so it encompasses all of the lightest pixels in the body. I can move this yellow one around. I can move it left or right, which is going to change which colors are assigned to the different pixels. I could drag this black dot at the far left, drag it way over to the left. What that's doing is it's causing this red dot here now to apply a red color to a lot of the relatively dark pixels. So a lot of things like musculature, some of the skin and stuff is starting to show up. The other thing that you can control with this is not just the position of the dot from left to right, but also the height of the dot. The height of the dot determines the transparency or the opacity of the objects that are colored with that color. So if I drag this red way up high, it now makes everything that's colored red, completely opaque. It's no longer transparent anymore. So we can actually see an image of the outside of the skin. In fact, looks like we're even seeing some straps or something that are holding this person on the table. I can drag the black line or the black dot over to the right a little bit and those go away, but we still see this perfect image of the anterior part of the person's abdomen and thorax. But if I drag that red dot back down, then all of those red pixels start to become more transparent. The skin goes away. Depending on exactly where I position this red dot, I can start to see things like abdominal muscles, like there's the rectus abdominis again, and the external obliques over here on the sides. I can use this to try to make certain things visible that are otherwise usually pretty challenging to see. Like, for example, I'm going to crop away front part of the thorax here, crop into the thoracic cavity a little bit. Like, let's say I wanted to be able to see the lungs. Well, it's really hard to see the lungs because they're almost black. And if we make all the black pixels visible, then that's just going to make all the air around the body visible. So this is where you can make some very subtle tweaks to make maybe the lungs visible, but not have the air around the body visible. So what I'm going to do is I'm going to take the, whoops, come on, where'd you go? There it is. We grab that black dot, drag it farther to the left. I'm going to add another dot somewhere over here. You can simply click on the line and it'll add a new dot for you. Drag that up. And notice how the lungs kind of look like they're starting to fill in with black or some very dark color. If I double click the dot, it brings up my color picker. I can pick something like, I don't know, like a bright purple. And it's now assigning that purple to the airspace inside the lungs. And then I can, again, play around with exactly where these different dots are located. To try to change colors and opacity of some of these different tissues. So I've got at least some purple showing up in there. The lungs aren't completely filled in solid, but we're definitely getting a little bit more value and a little bit more color. So you can, make some interesting colored things, and like I say, you can tweak these things to try to get certain tissues or certain structures to show up that would otherwise be difficult to see. Now, at the moment, I'm kind of stuck with this color scheme that I just created. But if I want to go back to the default color scheme, there are three little pop-up menus here. There's one called CLUT, and right now it says that I'm using my custom 16-bit color lookup table. But if I click the little triangle next to that, there's actually a whole bunch of preset colors that I can choose from in here. Some of these are really bizarre, like, okay, what's that good for? I don't know. But the default one that always comes up at the beginning when you open any data set is this thing called VR muscles and bones. So if you want to return to just the default color settings, you just choose this VR muscle and bone, and we're back to our original set of colors. And then we can, again, adjust, by dragging left and right, up and down to make different tissues visible or invisible. Oh, by the way, one thing I forgot to show you when I was in the 16-bit color lookup table editor, let me go back in there. I can now demonstrate exactly what that window level and window width tool is doing. This is the little grayscale icon tool. When I drag it left or right, it compresses. Look what's happening to the little color graph at the bottom. If I drag to the left, it compresses that little graph. So it's assigning color to a much narrower portion of the range of gray values. If I drag to the right, the opposite happens. It's stretching my graph. So it's now assigning colors to more of the pixels in the image. If I drag up and down, it's going to slide that whole color graph to the left or right. If I drag up, drags it to the right. If I drag down, it drags it to the left. So it's got the same colors, it's just assigning those colors to a different set of pixels. So, again, visually, you can kind of see what's happening. Drag left, scrunches up the graph, drag right, it stretches it out, drag up, drags it to the right, drag down, drags it to the left. So that's what that window level, window width tool is doing. Now, if you do something like this and your image just goes away, it's like, oop, I moved too fast, I moved too far. You can also go back to WLWW, that stands for window level, window width. Choose default. And all right, brings you back here, which is kind of ugly, but at least we got something. There we go. Okay, so the second half of your exercise today is to open up some other data set, turn it into a 3D volume rendering. And I think I gave some guidelines, like I think I wanted you to play around with the color lookup table editor and play around with the window width, window level, maybe even crop it a little bit in some plane, and then just export another JPEG throw it up on my courses. So there are two drop boxes up on my courses. One for you to upload your surface rendering, another one for you to upload your volume. And if you need a reminder about exactly what to post and what the parameters are that we're looking for, it's all posted in an assignment file in my courses. Okay, so feel free to take a break if you want. Sorry I talked so long. And I'll be around to troubleshoot. And Winnie is here in the back too to help troubleshoot. Oh shoot, I forgot about that one. Sorry, don't go anywhere. Yep, I always forget to warn people about this. Somebody said it's spinning on its own. That's right. This is one of the stupidest things that anybody has ever programmed into a piece of software. Go to the Horos menu. Settings. Horos settings. Click on where it says display 3D, the little 3D head. All right, I've already turned this off, but by default, when you do that, this will probably be checked. Auto rotate objects after 3 minutes of whatever it says in activity. It's like, why in the world would you ever want it?
That's such a stupid option.
Yeah, and it's turned on by default.